we've just started the nervous system, and the nervous system is specialized to transmit and process signals. Transmitting signals in the nervous system is a pretty complex process, so I wanted to give you a strong foundation in the structures that allow this to happen. So when you look at a neuron, neurons kind of look like a fried egg that has branches coming off of it. Okay, and in biology, often when you see branches, they are called dendrites. So these branches right here and here and here and here and here and here, these are all dendrites. And so dendrites that are on a neuron take in information. Okay, the cell body, which is right here, also takes in information. And here's our nucleus. You'll get a lot more structure in the lab portion. I just wanted to give you some anchors. So information comes in various parts of the dendrites and the cell body. Okay, so information is going in. So this right here is the receiving end. Okay, and this big branch is called the axon. Okay, and the axon is the end that sends signals. Okay, so right here, all the way down to these kind of axon terminals, this is where transmission happens. Okay, so information comes in on the cell body and the dendrites and goes out the axon. Okay. Before we dive deeper, I'm going to add that here on this side, where the dendrites are, chemical signals are going in. And here, electrical signals are being transmitted. Okay, so the chemical signal comes in, and it actually gets converted into an electrical signal. And that electrical signal is going to travel down the axon to the axon terminals, and at the end down here, it becomes chemical again. Okay? So I hope that this makes sense. I know there's lots of moving parts here. But chemical is coming into the dendrites and the cell body. It transitions into an electrical signal which gets sent down the axon, and then it converts back to chemical here. And where are these axon terminals sending signals? Well, they're sending signals to the next neuron's dendrites. Okay? It can also be a muscle cell or a gland, but right now we're just talking neuron to neuron. You may be wondering now, how does a electrical signal turn into an electrical signal and then turn back into a chemical signal? In order to understand how that happens, we need to look at some structures that are found in the plasma membranes of a neuron. And to be fair, these structures are found in the plasma membranes of many cells, but here they're specialized to send these signals. So hopefully you remember from Bio 107 that the cell membrane is made of a phospholipid bilayer. And so here's the phospholipid bilayer. And every once in a while you see these protein channels. Some of these protein channels are always opened, and those are called leakage channels. Okay, so whatever goes through those channels can just kind of bounce around and eventually hit the opening and go through. Other channels are called gated channels. And gated channels need to be opened. And there's 3 different ways that gated channels can be opened, and we'll look at that shortly. Before we look at gated channels, I just want to remind you that the key thing about protein channels is that they only allow certain substances through. So they are selective in that they will only allow molecules or ions of a certain size and charge through. So a sodium protein channel only allows sodium through. Same with potassium, leakage channels, etc. So it doesn't matter if it's a leakage channel or a gated channel. All of these channels are only going to allow a very specific type of ion or molecule through. Gated channels, as I mentioned, can be opened through 3 different mechanisms. There are chemically gated channels, there are voltage gated channels, and there are mechanically gated channels. Chemically gated channels have a mechanism that's familiar because it's similar to how an enzyme and substrate interact, that locking key mechanism that you heard about in Bio 107. So a chemically gated channel is going to have a receptor region, or an active site, I should say active site, that will fit a specific chemical. And so when that chemical comes along, so we have this little pink chemical and it's floating around, bouncing through the extracellular fluid, and boop, it bumps into that active site. And once that happens, just like in the enzymes you learned about in 107, it's going to change the shape of the protein. And it changes the shape of the protein in a way that opens this gate. It doesn't actually erase the gate. So, I'm erasing it just because I only have my limited art skills available to me. But in reality, it just changes the shape of the protein, and now it's going to allow substances through. Okay, so substances can go that way or they can go that way. Many channels are bidirectional. So substances can go either way, but the overall movement tends to go down a concentration gradient, right? So from high concentration to low concentration. And if that doesn't sound very familiar to you, go to Bozeman Science and watch his diffusion and osmosis video, and that will hopefully jog your memory. Okay, so chemically gated channels, they need to have the correct chemical bind to the active site, which changes the shape, or another way of saying that, changes the conformation of the protein, opening it up, and then ions can move in and out. The next type of gated channel is a voltage-gated channel. And voltage-gated channels are going to open in response to changes in voltage. So if normally this channel is closed at, say, negative 70 millivolts, it might open when the voltage is negative 55 millivolts. So again, a voltage-gated channel is going to open and close in response to voltage. So here we have the open voltage gated channel, and I put a little lightning bolt over here to help you remember that voltage gated channels open or close in response to changes in voltage. The last type of gated channel that we're talking about here are mechanically gated channels. And I'm not going to spend a lot of time on them because they actually don't come into play right now. We'll look more into them when we study the peripheral nervous system. But a mechanically gated channel is going to open in response to pressure. So here's the channel closed, and then we have a hammer that's hit the channel, and the channel has opened. My little drawings are just to help remind you what is happening that opens these channels. All of the channels that we've talked about so far are passive channels, and a passive channel does not need ATP in order for ions to move through them. The last of the channels that we need to talk about is the sodium-potassium pump. And the sodium-potassium pump is a pump that requires ATP, so it's called an active pump. It needs energy in order to allow the ions to move through it. And so it pumps both sodium and potassium, but it pumps sodium and potassium against their concentration gradient. So all the channels that we've been looking at are passive channels, and they just allow whatever ions or molecules are going to be going through them to go from higher concentration to lower concentration. But the sodium-potassium pump, because it requires energy, is able to pump sodium to higher concentration and potassium to higher concentration. Here we're going to organize our information about channels. So channels can be passive or active. Passive channels do not need any energy in the form of ATP, but active channels do. In addition to whether or not they require ATP, passive and active channels differ because in passive channels, the net movement moves down the concentration gradient. So overall, ions are going to move from high concentration to low concentration with passive channels. And in active channels, ions can move against the concentration gradient from a lower concentration to a higher concentration. So a good example of an active channel is the sodium-potassium pump. Passive channels can be further broken down into leakage channels, which are always open, and gated channels, which only open under certain conditions. Leakage channels are always open, and they can be added or removed from the plasma membrane. So if more water needs to come into the cell can insert more aquaporons in the cell membrane. And if less water needs to go through the cell membrane, it can remove aquaporons from the cell membrane. Gated channels are channels that only open with a specific stimulus. So that might be chemical, voltage, or mechanical, as I went through in the earlier examples.